Thank you, Trevor. So good afternoon, everyone. I'd like to thank everybody that's here today. I noticed that there's some new people in the audience. And my name, once again, is Chen, and I'm affiliated with the Center of Excellence on Health Disparities. And my topic of the talk today is omega-3 fatty acids and their role in model membranes, potential links to beneficial health outcomes. So the purpose of my study is to study the distribution of omega-3 fatty acids in model cell membranes. The reason why we want to study omega-3 fatty acids is because they are connected with multiple health benefits, such as cardiovascular health. Multiple studies show they compare populations that intake a high amount of fatty acids and they compare it to a normal diet sort of thing, and they see that the people who intake a high amounts of fatty acids actually have lower rates of cardiovascular disease, which makes us assume that they're improving their cardiovascular health because they're intaking high amounts of omega-3 fatty acids. But on the other end, there's a study that shows that if you intake too much or too little amount of omega-3 fatty acids, you have a chance of postnatal growth retardation, which is something we don't want to have, which is why we are looking at this study today. Furthermore, we want to take a look at omega-3 fatty acids because they're highly marketed. Everyone's throwing out this idea that omega-3 fatty acid is a healthy fatty acid. However, they don't even understand how once it is intaken and it's digested, what happens. So we want to take a look into which is why we ask the questions, how does omega-3 fatty acids get transported into our body and incorporate into cell membranes? Furthermore, one specific type of omega-3 fatty acid that we want to look at is DHA and how that affects the composition of our cell properties. And hopefully this molecular level study can help us take a look at the overall view of how omega-3 fatty acids affect our health. So the cell membrane, it consists of a lipid bilayer. And this figure up here is a simple model representation of what a lipid bilayer would look like. And we see here a molecule representation of a phospholipid. And we can see that this phospholipid has a head group and a tail group. And this tail group is what we want to focus on because that is the fatty acid group. And another thing to point out is that there are multitudes of head groups and multitudes of tail groups. Tail groups can be seen as really straight molecularly, or they can be seen as very bendy. And the unsaturated double bonds is what makes or allows for the tail groups to be really bendy. So what exactly is an omega-3 fatty acid? An omega-3 fatty acid is a subgroup of fatty acids where the first unsaturated bond is in the third position from the omega carbon. This is an example of an omega-3 fatty acid because if we count it out from the beginning, we see that the double bond happens right here in this third position. Furthermore, the study by McClements and Decker shows that omega-3 fatty acids play a major role in membrane fluidity, cellular signaling, as well as gene expression. So the specific type of omega-3 fatty acid that we want to focus on today is DHA. And this is an omega-3 fatty acid that contains 22 carbons and 6 double bonds. And furthermore, we can see that it's in high contents and variety amount of foods, and one type of food that we can see it's a lot in is fish oil. Furthermore, when we take a look on the cellular level, we see that there's a high concentration of DHA in sperm cells, brain phospholipids, and the retina, or the eye cells in your retina. This right here is a molecule representation of DHA, or a di-DHA, which is a two-tailed DHA molecule. And we can see it has 22 carbons and 6 double bonds. And then this representation right here, this is another model representation of what DHA would look like. So in the study, we use GUVs, or giant unilamellar vesicles, in order to observe how the incorporation of omega-3 fatty acids will affect the cell membrane. And we grow GUVs because they mimic the cell membrane. Lipids, this right here is an observation made under the microscope of what a GUV would look like, and this is a horrible picture, so look over here on the TV. And this is the molecular representation, and we can see that there's water on the outside as well as the inside of the vesicle. So we grow these GUVs by using electroformation, and basically what we do is we create our solution, and then we dry those solution onto ITO slides, or idiom tin oxide slides, which is just a fancy name for a slide that can conduct electricity. And we dry the lipids to evaporate all the chloroform, and then we hydrate the lipids in this chamber set up here, where we have the Teflon ring and then the water in the middle, and then we clamp it shut and it looks like that in the end. And then we electroform it in an incubator at 50 degrees Celsius with our desired parameters. And in this case, my desired parameters was a sine wave using 5 Hertz and 2.5 voltage. And what happens when we are setting the voltage through the chamber is we create an electric field. And when we create an electric field, there's a vibration that sort of allows the liquid to flow up and down, and the lipids will begin to bud off of the ITO slides, creating vesicles. And we observe the vesicles using fluorescence microscopy. This picture right here, or this figure right here, once again, it's really bad, so look at the TV, shows a cluster of GUVs made with the concentration of 5 to 5 to 90 molar percent ratio of DHA, cholesterol, and DPPC. DPPC is basically a saturated lipid, so we wanted to see how DHA, the unsaturated lipid, works with the saturated lipid. And we can see a nice close-up picture down here. From these images, we can see that we're able to create the GUVs and actually form the GUVs. However, we don't see lipid domains, or like patches of where the membrane shows different composition. And what you can see is like lighter areas and darker areas. This study that we took a look at showed these different patches. Therefore, we can see that it's, this is the sort of thing that we're looking for, but we may or may not be able to see them because they might be at a nanoscale. And if they are at a nanoscale, we need to use a different technique. Another technique that I'm looking into is atomic force microscopy, where we basically use a tip, like a nanoscale tip, and it scans the surface of your membrane, and it reads it out, and you get this nice topology, which, once again, you can't see here. And it shows different variance in heights, which will represent the domain areas. So some things to consider in this whole entire project, since it is an ongoing project, is the parameters at which my GUVs are made at, such as incubation time, frequency, voltage, and solvency. If we are able to change these, perhaps we're able to grow the GUVs, since a couple of my samples have been unsuccessful and they haven't grown. Furthermore, I want to start using the AFM and compare the results from the AFM that I get under the fluorescence microscopy. And another thing is that I want to compare different types of omega-3 fatty acids, because right now I'm only looking at DHA, and there's a lot of different types of omega-3 fatty acids, such as ALA and EPA, and those are also highly marketed out in supplements. So before we move on to questions, I'd like to acknowledge Professor Hirst and Kai Lohr, because they are the ones who actually guided me through this whole entire process and helped me do the research, as well as friends and members friends and colleagues from the COE as well as members from the Hirst lab for helping me sort of create the presentation and sort of get through all of this. And don't forget our funding agencies, because it wouldn't even be possible without them. So, any questions?
I have a question. Now, you said you made vesicles out of the DHA?
Yeah, we would basically make vesicles out of different lipid compositions.
Okay, I wasn't quite sure what you then observed from that.
Okay, so this is an example of the vesicle, right? And what we're trying to observe that we can't, we don't see on this picture, but we see it here, is like, you see how these lighter patches and darker patches? That's what we're trying to look for under the fluorescence microscopy, because that'll show the arrangement of, like, they decide to arrange in different locations and create domains.
I know you said that too much or too little can lead to side effects. So are you possibly looking at like how much would be too much or how much would be too little?
I wanted to start on this micro scale level and then work out, and eventually I would get to how much is too much and how much is too little, but that's like way out in the future, and it's going to take time to figure that out.
Stephanie, how specific is that chamber able to make those lipid vesicles in relation to their size?
Okay, well, the chamber itself is just the equipment that we use. It's not the chamber that makes the lipid vesicles per se. It's when we electroform it. So it's when we put it through, when we put a voltage through it. That's when we can see the vesicles start to form and bud off. And one thing that actually might change how big the size is of the vesicle is the voltage power or the frequency that we put through it. That might change the size of the vesicles.
Have you been able to play around with that? Or?
Yeah, I've been playing around with a lot of different things.
One more question. One of your potential benefits was, or one of the benefits that you said you get from omega-3s was, I think it was decreases psychopathic disease or something. Or also,
Are you talking about this?
yeah.
Okay, so omega-3 fatty acids are that you can find high contents of it in mother's milk, especially DHA. And the reason for that is because we need it in brain development, and most of our brain development is done as a baby, as a child. So if we actually have the efficient amount of DHA, then you would have a decrease in psychopathic diseases, because psychopathic diseases develop in the brain, and it usually develops because of some sort of brain development functioning as you grow older.
And by psychopathic diseases, what do you give me an example of such?
I think schizophrenia would be like something that has to do with neurons.